I get this question a lot, actually. And as with any tool, I think that you just have to be clear on the question you're trying to answer. What any of these sensors answer is, what is the force being experienced by the molecular host? So just for example, early on, my lab put tension sensors in a class of cell adhesion protein called cadherins. And they faithfully reported on the force experienced by the cadherin. But there are lots of molecular connections inside of a cell, all hooked up in parallel. So if you're interested in, for instance, the mechanical forces that a cell is experiencing broadly, molecular force sensors may or may not be the most direct path to your answer. If you're interested in force transduction through a specific series of molecules, they can answer your question better than any other tool I know of. So after playing with these things for a while, I think that the name actually says it all. They're molecular force sensors. They report on the forces that molecules experience. There are other tools available for measuring forces at the cellular level that are more appropriate if that's the question that interests you.
So my first advice is to, one, think very clearly about why you want to measure the tension across a specific protein in a cell. All right, so it's not the same as traction force. It's not a way to get an estimate of the whole mechanical work your cell is doing in a way that might be incompatible with another technique. What it tells you is where one type of protein is loaded. And so you should probably need a pretty good reason to want to understand that. And then again, this is also why I now kind of think of it as an analogy to a post-translational modification in mechanosensitive signaling. It very much focuses on this idea that this is a technology that's not really compatible with stresses and strains and 3D bulk continuums. It's a molecular readout and should be used in that way, which is exciting because there's no other way to do that. And I think that that's what enables us to talk about things like preclinical screens and things like that. The second thing is FRET looks like normal microscopy, but it's not. You have to be a little bit better at quite a few things. You need to be a little bit better at molecular engineering to make the proteins. You need to be a little bit better at cell culture than some people to make sure your cells are doing the right thing and the proteins expressed at the right level. You need to be good enough at microscopy that you can align your images to like sub-pixel. You need to understand the image analysis and all these things that go into it. And none of those steps is individually hard. Putting them all together and knowing that you're putting them together right is also not that hard, but takes time. And so I think that's what a lot of people forget is they've done maybe immunofluorescence or some of the very basic microscopy. And they're like, oh, well, I've done that, so I can do FRET. And that's not quite true. So the worst thing about FRET is no matter what you do, you will get a signal. It's very easy to get a signal. It's very hard to know what your signal means. So there's usually a large number of controls you have to do, and you have to understand why they need to be done. Those controls are what we call biological, like the cell is expressing the protein properly. They're also like technical, which we mean that the sensor is behaving properly. So many of us who work in this field have written quite long reviews about how to do it. But what I've noticed is most people want to skip steps in those reviews. And I'm here to tell you that's a mistake. All of those things that we do, we suggest you do them for a reason, because they need to be done. And so that's one thing that this question did ask is what would I give to people to create a new sensor? And my advice on creating a new sensor is different than using an existing sensor. So if you're going to create a new sensor, I strongly suggest you consult with someone who's done so before. It doesn't necessarily have to be a tension sensor, but a FRET-based sensor, because what I'm saying is we can build them very quickly. But there's about 2 years of controls you have to do. And if any of those controls fail, you have to start over. Because you have to pick a new insertion site. So it's a rather high risk, high reward endeavor. And so it's not something that you want to do on a whim. It's something that you want to do for a very precise reason, that you really need to know what the force is on this particular protein and why.
First advice, don't make your life harder than it needs to be, and talk to the people, to the labs that use these techniques already, and ideally to those labs that have a little bit of an experience with it. My sense is that in this field, people are very helpful, very willing to share their own experiences. And I think, young scientists that want to start in this area should just use that. As I said, people are helpful. Our own lab is always willing to share reagents and our expertise, also data analysis tools to help other research groups. And I think, you should be smart and use that. Second of all, I think first of all, before you start with these experiments, think again what your research question really is, and ask yourself, can tension sensors really help to answer the question that I have, to address my problem? Or are there other techniques that may be easier? Because tension sensors are not really easy to use. It requires investment in terms of infrastructure. It also takes a lot of time, takes a lot of careful data analysis. So really make sure that you have defined your research question sufficiently well and that your research question is specific. I think in general, we have to do this when we do scientific research, but with tension sensors, I would especially do that because it is a real commitment. And once you have decided to do that, I think the number one advice would be don't cut on controls. Don't make any shortcuts there. Rather make too many control experiments than too little, because you will need all of them to make a reasonable interpretation of your data. So controls, that's the number one rule when you're really doing the experiments.
This is a very exciting and emerging field of research. An investigator new to this field should keep in mind that this research requires highly interdisciplinary and integrative efforts from many different fields, including chemistry for sensor design and synthesis, biophysics and structural biology for understanding mechanodynamics, and molecular cell biology for biological and pathophysiological relevance. Hence, one of the most important factors for the success is building a strong collaborative team with relevant and complementary expertise. Related to this aspect, another point that I wanted to emphasize is that the importance of dissemination of newly developed tools. While many exciting biomedical tools have been developed by chemists and pioneers, poor accessibility to other investigators or incompatibility of these new tools to desired biomedical application hinders their broad applications. I hope that new investigator to this field considering dissemination of these new tools effectively.